All right, continuing where we left off, the next kind of special amino acid is histidine. Histidine, if you recall, has the capacity of being positively charged, so it can absorb a proton, so it's a base, becomes positively charged. What's interesting about this is it has a pKa of 6. So you don't know what pKa's are, so let me kind of explain. At a pH of 6, if you were to have 100 of these histidines, 50 would have a proton and be positively charged, and 50 would not have a protein and be neutrally charged. Okay, so it has the ability to flip back and forth. This pH is very close to physiologic pH, right? It wouldn't take much to nudge this forward and backwards. It also has another unique capacity in that when it absorbs that proton, it has the ability to slightly change structure. So take a look here at the protonated form where we have in the middle, and you can see that the only difference between this structure and this structure is here you have a double bond on top and a single bond on the bottom. Here you have a single bond on top and a double bond on the bottom. Okay? And this can flit back and forth between the two because the hydrogen is kind of balancing this pentagon ring. What's important here is because of this ability to switch back and forth, when it then loses that protein, it could take either one of these two forms, either form A or form C. What's important about this feature is we are essentially talking about an amino acid that can take a proton from one side, move it to the other side, and then release it. Okay? This can actually be part of the mechanism of how an enzyme works. So when we think about the active site of an enzyme, that site where you see actual chemical reactions being manipulated, you could see histidine in this site. As another example, when we have groups that have hydroxyls on it, such as serine and threonine, histidine could be in a position where it could deprotonate that hydroxyl, something that doesn't normally happen, but under the right conditions, histidine can pull it off. That would actually make the serine very reactive. Okay. Another thing that can happen to histidine under very unique conditions is it can also lose a second proton and become negatively charged. Okay? This requires specific circumstances for this to happen, but essentially we have an amino acid that can be positively charged, neutrally charged, or negatively charged, and it can take protons and move them from one side or to the other and release them. This can act as a powerful mechanism in the active sites of proteins. So consequently, one of the most frequent amino acids that we see in an active site is in fact a histidine. It's not the only, but this unique nature makes it a very versatile thing. Now, when we take various amino acids and try and link them together, we call those linkages, those covalent bonds, peptide bonds. And so a polypeptide is a polymer of amino acids. And polypeptides can range from just a few amino acids to thousands of amino acids. Another thing to consider is that each polypeptide will have an amino group at one end and a carboxyl group at the other end. We refer to this as the N-terminus and the Z-terminus. So let's take a look at a diagram of this. So here we have 2 amino acids already in a polypeptide. So here's a peptide bond. Here's an incoming amino acid that's going to be linked. You can see this is a dehydration reaction. So we remove water and we get this carbonyl carbon linked to this amino group, resulting in a peptide bond. Something that's interesting about this peptide bond is we get free rotation around either side of this carbon. So here's the alpha carbon. We can get rotation around here and rotation around here, but we do not get rotation around this peptide bond. So whenever you have a peptide bond, this forms a kind of rigid structure. And that limits how much this backbone can bend and helps determine its overall shape in the future. And we can talk about why this is in class. Now, the real key to a protein is that it has a three-dimensional shape that results in some specific function. You can see that the internal shapes of these proteins can be very complicated. So we have different ways of representing these proteins. So here we have something called a ribbon model. The ribbon model highlights various what we call secondary structures, which are these flat parts here, these arrows, and these helices. And it highlights how they are overlaid or folded into each other. So this really looks at how does a protein fold. This doesn't really tell us what the protein looks like. What's better for that is a space-filling model. In this space-filling model here, we can see that it's one big solid gunk, but it has this groove, right? When you have things like grooves, you oftentimes have some type of binding that might happen in that groove. Here's a combination where we get kind of a ribbon structure in the back that helps us follow the backbone, but we can also see all the R groups present in this little wire frame. And here we actually have the molecule that binds into that site overlaid. So we can kind of see how these various amino acids interact with its target molecule and what's important for binding. So we have lots of different ways of representing proteins. Now, something to consider is the sequence of amino acid determine the protein's three-dimensional structure. Okay? This is, again, directly related to gene structure, or the sequence of nucleotides in the DNA. Now, since this amino acid sequence determines the three-dimensional structure, we also need to keep in mind that the protein structure determines how it works. So the function of a protein usually depends on its ability to recognize and bind some other molecule. Okay, this is typically what's happening with a protein. It's either linking up with multiple copies of itself, or it's recognizing some target to be an enzyme on, any various different things. Here's an example of an antibody. Remember, this is a defensive protein. And here is a protein from a flu virus. You can see that here's the surface of one of the flu particle, and here is the surface of the antibody. They are completely matched, and this is how this antibody is capable of binding this protein from a flu virus, and consequently how the antibody binds the flu to begin with. Now, there are 4 levels of protein structure. So the primary structure is the easiest to understand. This is simply the unique amino acid sequence. Okay, so we can see here we have an amino end that has its N3 or NH3 positive, and it would progress on to its carboxy terminus. But it's just simply the sequence of amino acids. and this is directly related to the DNA sequence. The secondary structure is referring to various coils and folds within the polypeptide chain. So here there are a couple of structures that tend to form on their own, something called an alpha helix and something else called a beta pleated sheet. These structures depend upon hydrogen bonds within the backbone itself, and these structures tend to form independently. The tertiary structure is when you take all of these secondary structures and fold them into an overall three-dimensional shape. And we can look at it like this. Here we can see that there are some of these beta pleated sheets. We can see some alpha helices. We also see some loops occurring here and some sharp turns that might be occurring. This overall three-dimensional shape is its tertiary structure, and this mostly is what determines that protein's function. On occasion, we have proteins that don't work by themselves. They work with other protein partners. In that case, we can actually refer to a fourth level of structure that we call the quaternary structure, where we have multiple polypeptides coming together to form some overall structure. So here we actually have 4 copies of this tertiary protein that then folds into this even larger three-dimensional structure. All these protein levels of folding are related and are governed by different forces. So let's take a deeper dive into them. So the primary structure is completely dependent on its amino acid sequence. This means that the bond type that is important is covalent bonds. So its structure here is completely determined by the covalent bonds. The sequence of amino acids is completely dependent upon the information from the gene that governs it, and we'll talk more about that later. Secondary structure is a little harder to understand. So because of how the backbone works, because of the fact that the peptide bond cannot be rotated, but you can rotate around either side of the alpha carbon, this leads to a couple of structures that can form on themselves. One is called an alpha helix, and the other is a beta sheet. And these structures are completely dependent upon hydrogen bonds that stabilize it. And it is specifically hydrogen bonds within the backbone itself. So these structures are kind of independent of the R groups, or the side chains that exist in the amino acids. Now, there are additional structures that kind of fall into the secondary range, things such as turns, loops, and disordered regions. We'll talk about that here in a second. Let's zoom in here on an alpha helix and take a look at it. We'll rotate it around. So the backbone is completely dependent, or the helical shape is completely dependent on the backbone and hydrogen bonding within it. If we add in the amino acid side chains, what you'll notice is that all of the side chains point outward. Okay? This means that we can get these cylinders that have different faces on it. We could have one side of the cylinder be hydrophobic in nature. We could have another side be maybe hydrophilic in nature. This diversity can lead into the overall three-dimensional shape of the protein, or the tertiary structure. We will pick up with beta sheets on part 3 of this video.